Hi, my name is Anay Ravelo, and I am a PhD student under the advising of Dr. Luciano Cacheta. And for this video abstract, we will be discussing the association between hyperketonemia and rumen parameters in the first 2 weeks postpartum. So hyperketonemia is defined as the elevated concentrations of circulating beta-hydroxybutyric acid in the blood, and it is indicatory that adipose tissue is being mobilized as an alternative source of fuel. Subclinical levels of hyperketonemia are identified as greater than 1.2 millimolar per liter, and clinical levels are identified as greater than 3.0. When and how does this happen? Well, after calving, cows are in a state of negative energy balance, which is a result of decreased dry matter intake, since they do decrease their feed intake after calving, and increased energy demands for lactation since they begin producing milk. Thus, there's increased production of ketones that occurs in the liver and enter the blood circulation. Why is this a problem? Well, hyperketonemia is associated with decreased milk production, impaired reproductive performance, and increased removal from the herd, all three of which can be very costly to producers. The interest of this particular experiment is what is occurring in the rumen of cows with hyperketonemia. Previous studies have identified that there are changes in VFA in the rumen of cows with hyperketonemia, where they have decreased levels of acetate and propionate compared to control cows. The objective of this study was to investigate if there is a difference in the rumen environment of cows with and without naturally occurring hyperketonemia. The design of the study was an observational design that included 12 cannulated cows, of which were followed for 2 weeks after calving. We collected blood and rumen samples from these cows, and the cows for the hyperketonemic group were identified as cows that at any point during blood collections had a BHB concentration greater than 1.0 millimolar per liter. Although this concentration is lower than the classical definition of hyperketonemia, we allowed this in order to have more animals in our hyperketonemic group. Blood samples were collected to measure BHB, glucose, and NEFA, and rumen fluid samples were collected to measure BHB, pH, and ammonia. For the results for the blood parameters, we see that glucose, depicted here in yellow, was greater for the cows in the hyperketonemic group and decreased throughout increasing days postpartum. And for the control group depicted in red, we see that their glucose concentration was more stable. For NEFA concentration, we see that for the hyperketonemic group, it was increased around day 5 postpartum and then stabilized around day 12. And we see that for the control group, it remains fairly stable, similar to the glucose concentration. For results for the rumen fluid parameters, we saw no differences between pH and BHB, but we did see a difference in ammonia concentration between groups, where the hyperketonemic group had about 4 units greater ammonia concentration compared to the control group. For conclusion, our preliminary results indicate that there is a difference in ruminal fluid metabolites when cows have hyperketonemia. Thus, for future plans, we plan to analyze VFA, microbiome, and metabolomics of the rumen fluid samples collected. We also plan to collect more samples from the cannulated cows once they calve again. And we believe that identifying and understanding these differences could aid in implementing better treatment strategies for cows with hyperketonemia. I would like to acknowledge the guidance of my advisors as well as the help of my lab members in this project. Thank you.